Hi, Mr. Ernesto and friend. My name is Hamilton Guzman and this is my group. Let's talk about DNA function and structure, and also some important topics of DNA. There are: what does DNA look like? How are copies of DNA made? What are mutations? What is DNA? How was discovered DNA? And what is the role of DNA and RNA in building proteins? Thank you.
I'm going to talk about DNA. DNA is the genetic material that a cell needs to its growth and activities, and it also contains information to determine if it has an inherited characteristic. Scientists also describe DNA as a code. How was DNA discovered? Well, it was discovered not just in one night. It was discovered for many years by many scientists around the world. For example, in 1869, Friedrich identified a substance that later on will discover more. Then in 1919, Phoebus Levene identified that this substance contained 4 nitrogen-containing bases and phosphate groups and sugars. Then the 4 nitrogen-containing bases were adenine, thymine, cytosine, and guanine. Then, in 1915, Erwin Chargaff discovered that adenine equals the amount of thymine and cytosine equals the amount of guanine. Then, in 1951, Rosalind Franklin and Maurice Wilkins identified the structure of DNA using X-rays. Later on in one year, Alfred Hershey and Martha Chase discovered that DNA could change throughout experiments that they made. And finally, in 1953, James Watson and Frank Crick discovered the structure of the DNA, which was a long.
I am going to tell you about what does DNA look like. First, is a shape of DNA is double helix. DNA made of nucleotides. First, what is double helix? Double helix is a structure of DNA. 2 sides of leather often referred DNA backbone. The backbone are made of sugar and phosphate groups, and rungs are made of base pair of bases, each attached to one of the sugars in the backbone. Thank you. Then DNA made of nucleotides. The nucleotides are bases, sugar, and phosphate. The nucleotide has 4 bases. Which one? The bases are thymine, adenine, cytosine, and guanine. These bases from the alphabet coded. And a coded store information about which the protein the cells should building. The kind of protein cells in your body also determine your trait. Thank you.
Hello, Mr. Ramirez and friends. My topic is DNA function and structure. How are copies of DNA made? The copies of DNA are made by a process named replication. In replication, there are 4 steps during the copy to do the copy. The first one is when the 2 strands separate from the DNA. The second one is when the strands are used as a pattern for the new strand. The third one is when the nucleotides are added, and the final step is when replication finishes, when there are 2 strands equal of DNA molecule. When are copies of DNA made? The copies of DNA are made before the cell divides. It copies the DNA so each new daughter has a complete set of its What are mutations? Mutations are change in type or order of bases in a cell, in a piece of a cell. In mutation there are three kinds of change: insertion, deletion, and substitution. In insertion, a new strand are added to the base. A new base is added to the complete base. In substitution, a base is changed by another base. In deletion, a base gets out of the complete base. This is substitution. We can see here that this base was changed by a base of here was changed by this base. In deletion, this base was get out of this complete base.
I'm going to talk about the role of DNA and RNA in building proteins. There are two forms that contribute in the construction of proteins on a cell: transcription and translation. But first, we need to know how DNA and RNA structure are. This is a DNA structure. DNA is made of nucleotides. The four nucleotides are guanine, thymine, adenine, and cytosine. They are the constructing blocks of DNA. DNA backbone supports its shape. RNA is a molecule that is present in all living things and in their protein production processes. It also has sugar levels different for DNA, and it has a nucleotide named uracil instead of having one. Transcription is a process in which the information in DNA is copied to a messenger RNA. The steps of transcription are: 1. The DNA is taken as a support to make a new complementary strand of messenger RNA. Second, the DNA opens where the gene is located. Third, DNA complementary bases match up with the RNA bases. And finally, DNA closes and the messenger RNA is released. This process have the contribution of DNA, messenger RNA, and RNA. Translation is a process in which the information copied on the messenger RNA is used to build proteins. The steps of translation are: once the ribosome enters to the messenger RNA, transfer RNA delivers amino acids to it. Second, each group of 3 bases strand code for one amino acid. And finally, the genetic code determines the order in which amino acids are brought to the ribosome. This process has the contribution of the messenger RNA, transfer RNA, and DNA.
Hello dear friends and teacher. I will talk about what is the role of the DNA and RNA in making proteins. First, what is RNA? It's the necessary copy copy of the DNA. And there are 3 types. There are the messenger RNA, ribosomal RNA, and the messenger RNA. They are made of sugar phosphate bases, and the bases are adenine, uracil, guanine, and cytosine. And now I will talk about the processes. The first process is transcription. It's when the RNA made a necessary copy of the DNA, introduced the RNA and DNA. First, individual genes are transcribed. Second, DNA is used as a template to make proteins. Third, DNA express opens when gene is located. Fourth, RNA images RNA match up with the DNA template and then the RNA opens up and the DNA closes. Now I will talk about the translation. What is translation? It's the process of making proteins, and the first step is the mRNA is fed by a protein line. Second, it passes through the ribosome. Then the transfer RNA transfers the amino acids. Then the amino acids join together to form a protein.